Alright, so we are going to talk about the conversion of pyruvate to acetyl CoA. Now, if you know the acetyl CoA, this is basically a substrate or the starting substrate for the TCA cycle. And the pyruvate, it comes from glucose through the process of glycolysis. So there's this intermediate step where pyruvate is converted to acetyl CoA by the help of an enzyme complex called pyruvate dehydrogenase complex. Now, this is an enzyme complex, and it has got three different subunits, and we call that E1 subunit, which is the pyruvate dehydrogenase subunit. The second one is called E2 subunit. We call it the dihydrolipoyl acetyl transferase. And we have the E3, which is called the dihydrolipoyl dehydrogenase. Now, all these three different subunits, they work in tandem in order to convert pyruvate to acetyl CoA and release a molecule of carbon dioxide in the process. Now, there are two reactions that are going on. One of them is the dehydrogenation, and the other one is the decarboxylation. And because of this decarboxylation only, we are getting a molecule of CO2 for every pyruvate molecule that is going through this process, that is getting converted to acetyl CoA. So we're going to dig a little bit deeper into these three enzymes and look into how they convert pyruvate into acetyl CoA, one after the other. But before we move in, these three enzymes, they require different prosthetic groups or coenzymes to work, otherwise they cannot work. So namely, there are 5 different prosthetic group or coenzymes. I'm going to list them out over here. So coenzymes / prosthetic group required by these three, E1, E2, and E3. So the first one is thymine pyrophosphate. Short, we call it TPP. The second one is FAD, that is flavin adenine dinucleotide. Third one is coenzyme A. Fourth one is NAD, that is nicotinamide adenyl dinucleotide. And finally, the fifth one is riboflavin. Right. So these are the different coenzymes that are required by PDH, or pyruvate dehydrogenase complex, in order to work properly, in order to convert pyruvate into acetyl CoA. Now let's look into the process. So first we have pyruvate. Let's draw it like this. Structure of pyruvate: CH3, C double bond O, again a C. So this is the structure of pyruvate, and pyruvate gets releases this CO2 from here. It releases the CO2, but in the process, what it does, there's this TPP, that is thymine pyrophosphate, inside E1, that is pyruvate dehydrogenase subunit. In the process, this TPP, this gets converted into TPP, which is in which there is an addition of the rest of the group of the pyruvate that was remaining. So we have a hydroxyethyl TPP. So this is hydroxyethyl TPP, right? So it is giving off the carbon dioxide. We got the carbon dioxide of the process, and in the process this TPP is getting converted with the rest of the pyruvate which was remaining into hydroxyethyl TPP. Now this hydroxyethyl TPP, when it forms back to the normal TPP, what it does, it comes in contact with a lysine side chain amino acid which is present in the E2 molecule, or the E2 enzyme. It comes in contact with the lysine side chain, and this lysine side chain is having a lipoic acid group. So this is the oxidized form of the lipoic acid group, and when it comes in contact with, while it is coming back to the TPP form, this oxidized form gets reduced to the reduced form, and this part over here gets attached to this reduced form. So I'm going to cover this in yellow. If you're getting confused about this C double bond O, then let me draw it like this. This part, let's erase this. This is something like C double bond O. You have an H, you have an H. So this is getting cut off from TPP and it is getting attached to the lysine. I'm going to write it over here. Attached to the lysine, the side chain, the lipoic acid side chain of the lipoic acid modification of the lysine side chain, and this is getting attached to the reduced part of the lipoic acid. So let me write it down over here. This is the oxidized lipoyl lysine, and this is the reduced form, but it is acetylated, so we're going to call this the acetyl lipoyl lysine. So actually I'm drawing this lysine in the middle just to indicate that this lysine is getting converted into this. So there is only one lysine which is getting converted, not two lysines. Like, since I've drawn two side chains, it does not mean that there are two lysines. There is only one lysine which is getting converted. So now we have it, the acetyl form of the lipoyl lysine. Now when this acetyl form gets converted into the reduced form, so I'm going to draw an arrow, and again the same lysine is getting converted into its reduced form. So I'll draw it like this. Again the side chain, or the lipoyl side chain, and here SH, SH. So the acetyl group is getting deducted from the sulfur group. And when this is getting reduced, so let me write it down. This is the reduced form, reduced lipoyl lysine. So when this is getting reduced, what happens? There's this coenzyme A which has got an SH group. This coenzyme A is getting acetylated. So we have CH3, C double bond O, and S, CoA. So again, this part is coming from here. So this coenzyme A is getting acetylated to this, and now we call this the acetyl CoA. Right? So we got our acetyl CoA. Now what happens with the reduced lipoyl lysine? It cannot stay in the reduced form, otherwise the cycle won't continue. So what happens? This reduced lipoyl lysine, it gets converted back to this oxidized lipoyl lysine. In the process, it gives out electrons to FAD, converting it to FADH2. So it is reducing the FAD. And when FADH2 gets converted back to FAD, it gives out its electron, and it reduces NAD+. So NAD+ gets reduced to NADH plus H+. So the cycle is completed. So finally what we have our acetyl CoA, which we needed for the TCA cycle, and above that we have the carbon dioxide that is a part of the decarboxylation process. And if we mark the things according to the E1, E2, and E3, so this part, the one which I covered in this purple, this is the E1. I mean, this whole thing is going on inside the E1. Next, the E2 would be this part. except the FAD. So this part is the E2. So let me write it down. This is the E2. Finally, we have this part, the last part we have, this is E3. So like I said, the E1, it is called the pyruvate dehydrogenase. The E2 is called the dihydrolipoyl acetyl transferase. The E3 is called the dihydrolipoyl dehydrogenase. And if you look at the names, actually the things are happening. So in pyruvate dehydrogenase, so we are getting rid of this acetyl group and we are putting it onto the lysine side chain of the lipoic acid. In E2, we have the dihydrolipoyl acetyl transferase. So it is transferring the acetyl group from this TPP to the reduced form of the lipoic acid side chain on the lysine. And finally, in dihydrolipoyl dehydrogenase, again this reduced form is losing the hydrogens, giving it to FAD, making it FADH2, and getting back to the oxidized form of lipoyl lysine. So this cycle continues, and for every pyruvate that we have, so let me write this down. This is pyruvate. So for every pyruvate that we have, we get one molecule of CO2, one molecule of acetyl CoA. So from one glucose molecule, we get two molecules of pyruvate, and hence we have two molecules of CO2 plus two molecules of acetyl coenzyme A, and this goes on to the TCA cycle. So that's about it for this video, and I hope you liked it. I hope you understood the whole process, and I'm going to see you in the next one. Bye bye.